This has been a lot of fun listening to everybody's talks. I'm really excited to introduce our next batch of lightning talks this afternoon and our final batch as well. My name is Kim Popek. I work at Mount Rainier National Park I do ecological restoration work here, so I supervise the native plant restoration work here at the park as well as invasive plant management. So the title of this next round of lightning talks is Public Lands. So the brief description for that is, with growing visitation, compounding outside stresses, parks are grappling with novel changes around rising human use. At the same time, new tools are emerging to understand these effects and restore impacted areas. How can social science guide communication for resource protection while welcoming a broader coalitions of stewards? So really excited to introduce this next batch of presenters and presentations. Really excited to listen in. Our first presentation is going to be from two botanists with the Siskiyou BioSurvey group. John Villella and Jason Clark, but I'm not sure if Jason Clark looks like he's joining as a presenter right now. So maybe just John. But the talk's going to be about the effects of visitor use on small scale plants at Mount Rainier National Park. Really excited to hear about it. Thanks, John.
Sharing my screen here. Okay, can everybody see that? Okay, well, thank you very much for having me. My name is John Villella. I'm a senior botanist with Siskiyou BioSurvey, and I'm here to talk about the results from a study that we did in Mount Rainier National Park, looking at the small scale plants, the lichens and bryophytes in that area. So just to kind of give you a bit of some background here, the organisms that we focused on in this study are the small statured plants, including lichens, mosses, and hepatics, which are also known as liverworts. These plants can be seen throughout the park, growing on the sides of trees and shrubs, on rocks, and on soil. We know that they contribute substantial biodiversity to the park, and we were curious to know if they were being impacted by visitor use. We were particularly curious about the impacts from trampling, for instance, from visitors walking outside of designated trails. So the areas that we focused on for our study, we wanted to compare high use areas in two different ecosystems within the park. The high elevation alpine areas and the lower elevation forests. For the upper elevation areas, we focused on Sunrise and Paradise, and for the lower elevation areas, we focused on Ohanapakash and Carbon River. And this slide shows the locations. This slide here shows some of the methods that we used, diagrams of the various methods. We recorded the impact in areas of high visitor use and areas where we assumed the visitor use was more limited. These included random plots, belt transects across trails, and intuitively determined plots. Because of the small stature of a lot of these organisms and their very microsite requirements that might not be captured in random plots, we conducted also a number of intuitive plots and at areas where we discovered rare plant species in order to try to capture the diversity of the local hotspots. So for the results here on the biodiversity aspect of it, we documented almost 500 different species, including 24 rare species, 2 species which were not previously known from Washington State. 11% of the species that we documented are known from fewer than 6 sites within the state, and 13% were documented are known from fewer than 20 sites in Washington. We also found 8 species that have fewer than 6 global occurrences and 9 species that have fewer than 20 worldwide occurrences. Here's pictures of some of the species that we encountered. So hopefully by this you can understand that there's a rich diversity of these organisms in Mount Rainier National Park. Okay, so in terms of the visitor use impacts from trails, the on this slide, you can see 4 graphs showing the impact of visitor use on these communities. The left column is the alpine sites, and the right column is the lower elevation forested sites. With the graphs on the top showing the results from each of the different groups, so lichens, mosses, and hepatics, and the lower graphs show the results from the different substrate groups, and so that would be which type of what it was growing on, either rocks or soil, etc. Along the y-axis, you have species richness, and along the x-axis, you have the distance from the trail. And focusing now on the top graphs, you can see that in both the alpine and the low elevation forest, species richness increased as you move out from the trail. In the lower elevation forest, the increase in species richness levels off at 4 meters, but continues to increase as you go out further from the trail. In the alpine areas, there's a steep increase going out from the trail, and it continues to increase the further you get out, indicating that visitor use impacts are felt by these small plant communities even relatively far distance from the trails. In terms of the rare species that we encountered, in the alpine areas, there were several factors that are significantly correlated with the rare species occurrences. Rare species populations were located in wetter, rockier, and less trampled areas. In lower elevation forests, the rare species, the populations were more likely to be found further distance from the trail, but none of the other measured factors to be significant drivers for species occurrence in the low elevation forests. So in conclusion, our study shows that the impacts of visitor use can extend great distances from the trails anywhere in the park. And also we suggest that in order to preserve populations of rare plants, rare bryophytes and lichens in alpine areas, managers should focus on preventing the visitor use or directing visitor use away from wetter, rockier, and less trampled areas where the rare populations are more likely to occur. And here I've got a shot of the team that worked on this, and I'd like to thank them and also thank Beth Fallon for inviting us to do this project in Mount Rainier. Thank you very much.
Awesome. Thanks, John. That was really interesting. Amazing to hear just how many species we have out here in the park. That's really cool. I'd love to learn more. Thank you. Up next, we have Lindsey Skidmore joining us today from the University of Washington. She worked with us out here at Mount Rainier this last summer, and also kind of a similar theme here, assessing the impacts of off-trail hiking on subalpine meadows at Paradise in Mount Rainier. Thanks a bunch, Lindsey. Excited to hear what the results were from this summer.
Hey, Kim. Hi, everybody. I'm Lindsey. And today I'm going to be talking about my project, Remote Sensing Analysis of Social Trails in Subalpine Meadows at Paradise Mount Rainier. Social trails are unmaintained trails that are caused by people walking off of the official trail system. They often happen when people try to avoid water, mud, or snow on trails. There are too many people on a narrow trail, hikers don't know where they're going, or when people leave the trail to take a photo or rest. Paradise, home to many of Mount Rainier's iconic subalpine meadows, has fragile vegetation, which have very short growing seasons since they're snow-covered most of the year. Wildlife depend on these meadow habitats for survival, and in recent years, with an increase in vegetation, there are more people going off-trail and damaging these essential ecosystems. For the social trail probability model, we used high-resolution snow-free imagery and field data collected over the past 2 summers in a random forest algorithm. The prediction model shows likelihood of an impact for each pixel at Paradise Meadows. The model is then field validated during the summer months by confirming the model's accuracy to predict an impact for 200 random 50 points in Paradise. This is the final run of the model, side by side with high-resolution imagery. Light yellow indicates a high likelihood of disturbance, and purple represents a low likelihood of disturbance. The final field accuracy for all 250 random points was 53%. There was a couple reasons why this model did not perform as well as we'd hoped. First being overprediction in rocky and barren areas was one of the biggest reasons for a low accuracy. The model struggled to distinguish between rocky areas and bare areas and areas that had been trampled by visitors. Image resolution availability limits our ability to see impacts. Even at 25 centimeters, as we see here, it's challenging to see impacts in aerial imagery. At 60 centimeters and 91 centimeters, these impacts are even more difficult to see. Super high resolution, better than 25 centimeter imagery is expensive to get and needs to be captured for a very small window when the meadows are snow-free and are green to see impacts the best. Social trails can just be hard to see from aerial imagery in general, though. Shadows, trees, and small trails can make it hard to detect impacts from above. Field data collection is a critical part to training the prediction model and creating an accurate inventory of impacts of paradise. For the past 3 years, field data collection has been scientists and parks interns hiking around paradise to precisely inventory visitor impacts. This process is time-consuming, with one intern tasked each summer to improve on the accurate and detailed inventory. Over the past 3 summers, much of paradise has been mapped, but there are still many areas that have little or no data collected. Because of the inherently time-consuming nature of field data collection, impacts are happening faster than can be recorded. Some areas that were fully mapped 3 years ago might look completely different next summer as a result of increased trampling or restoration. Field data collection season is only a few months of the year. Depending on snow, collection at paradise can be from late June to September, which is not very long. Also, barren alpine areas have little vegetation, making it hard to distinguish social trails. These high elevation areas are often snow covered, the longest, meaning that they're only snow free for a few months of the year. Mount Rainier is a rare and special place that people want to visit, connect with, and recreate in. People want to see the iconic meadows, rivers, forests, and glaciers that Mount Rainier is so loved for. To preserve these habitats for future generations, there's a need to find a solution for sustainable visitation. Mount Rainier and Paradise Meadows in particular are at risk of being loved to death. With a steady increase in visitation each year, there is more human pressure on these ecosystems now than ever. With the current inadequate accuracy of remote sensing models, we need to continue to collect an accurate field inventory of impacts to assess meadow condition going forward. Remote sensing models may be feasible in the future when high-resolution imagery or high-density LiDAR become available for paradise. Looking forward, these are some of the strategies that have been implemented to help limit and record visitor impacts on subalpine meadow ecosystems. Timed entry was piloted last summer and helped to reduce trail congestion and pressure during some of the busiest days of the year. Continued monitoring and recording of impacts at paradise and expanding data collection at sunrise will be critical to assess meadow disturbance over time. We can continue meadow management, including restoring disturbed sites and using physical barriers like rope and rebar to keep hikers trail. Lastly, continuing education on why going off trail is harmful is essential to keeping Paradise Meadows the special place we all know it to be. And a huge thanks to all my collaborators from Mount Rainier and at University of Washington.
Awesome. Thanks, Lindsey. Beautiful pictures, by the way. Some of those were gorgeous.
Thanks.
But yeah, thanks so much for presenting, and really awesome to hear about your work up here this summer. Thanks a bunch. Okay, our next presentation is from Deneb Maine, who is a graduate student at Miami University and worked with the Dungeness River Nature Center. This is about monitoring the restoration of the Hurricane Ridge septic meadow. Thanks a bunch. Looking forward to hearing about it.
Okay. Hello, everyone. I'm Deneb, and this is my project on monitoring this restoration site. So my research is taking place up at Hurricane Ridge. So here I have a map of Western Washington with Hurricane Ridge pinpointed. And Hurricane Ridge is one of the easiest access points for high country access to Olympic National Park. So it ends up being sort of a key location for that. So in the summer of 2020, because it is such a popular access point, they did need to redo the septic system. So on the left here, I have a picture of them in the process of doing that, and which involved digging up about 5 acres or so of the subalpine meadow there. Throughout that summer, vegetation crews did as much as they could to salvage all the sod and very carefully replant it, which you can see on the picture on the left. In addition to that, the restoration also involved planting plugs grown up in the park. About 30,000 plugs were planted, and I have a picture here of several of those freshly planted plugs. So that then brings me to my big research question. And I wanted to know, like, was this restoration project successful? And specifically, I ended up looking at both percent vegetation cover as well as the species composition. And on the top here, can see a photo of the restored septic meadow with a transect line running through it. And on the bottom is a picture of the surrounding reference meadow that I had with a transect line and a quadrat. So what did I find? This right here is a chart of the percent vegetation cover. And in terms of vegetation cover, it was actually really exciting. It seemed like there wasn't much difference between the two. And vegetation cover is pretty important in terms of erosion control. So that was good to see. And then what I think was an interesting piece of this really was looking into the species composition. So the picture on the left here, I have mountain sandwort, which is by far the most common, like, dominant species in the surrounding reference meadow. It pretty much, like you'll see in the next chart, the most common one that I was finding. But it was pretty sparse in the septic meadow despite being heavily planted. And then on the top right, I have a quadrat with some yarrow. In the septic meadow, the yarrow was by far the most common plant that I found. And while it exists in the surrounding meadow, it is pretty sparse and sparse enough that my data didn't actually capture any of it. And then the other thing I found was in the bottom right corner, there's a quadrat with some grasses, specifically poas. And there seem to be a couple species. We're still working on some of the details of the ID of various potentially introduced grasses based on the growth patterns. Seems to have possibly been introduced with some of the nursery stock and will be something to keep an eye on. They were not seen in any of the surrounding reference meadow. So here I have a chart for the species composition. I did that via a point intercept, and on the y-axis I have the frequency of the points that I found, and these are the most common species. There are a few more, but they were sporadic enough that didn't make as much sense in terms of the statistics analysis. And so this then actually comes to the really big, key, important questions of what are the next steps. I do plan on continuing this study over the course of the next couple of years, hopefully, to start getting some of that over time details, because this data that I have was only just from this past summer, with some long-term hopes and dreams and goals of this actually turning into a much longer term, being able to see the long-term dynamic patterns through hopefully being able to turn this into a community science monitoring project. As well as it would be interesting to look into if any of these differences in species composition that I found, if those actually have an impact in terms of the ecosystem function. Okay. And then that does lead to my final conclusion of thank you very much, and definitely need to provide lots of acknowledgements. Many people, including some of the current park staff, as well as past crews, and all my classmates and instructors were very useful. And on the right, I do have a picture of a bunch of us helping out with that restoration project in 2020.
Awesome. Thanks so much. It's so encouraging to see successful restoration projects like that. So that's really neat. I definitely have some more questions that I'm excited to ask during our question and answer session at the end. Which also just a reminder to everybody, as we're going through these talks, please feel free to throw those questions in now. And then when we get to that questions and answer session at the end, we'll get to those questions. Okay, our next presentation is going to be Jill, who's going to be joining us. She's the executive director of the 10,000 Year Institute, and we'll be talking about assessing and addressing effects of invasive reed canarygrass on aquatic habitats, cutthroat trout, and coho salmon in Olympic Peninsula watersheds. Thanks, Jill.
Thanks so much. I just shared my screen. I hope it's visible.
It is. We can't see you yet, though. Is your camera on, Jill?
My camera is off. Sorry, let me turn it on. We're on slow-mo here in Forks, and
we're good. No, everything's good now. Perfect.
Great. Hi, everyone. Great to be here. And I am here to present on the reed canarygrass work that we've been doing in partnership on the Olympic Peninsula for the last 15 years, assessing and addressing effects of reed canarygrass on aquatic habitats, cutthroat trout and coho salmon in Olympic Peninsula watersheds. And I'm co-presenting with Bob Vadas from DFW, who's been doing amazing research up in Irely Creek, Irely Lake, and Big Creek in the Upper Quinault. And so he might pop in at some point. And let me see if I can advance my slides. I guess I need to go to slideshow, right? Am I in slideshow? No.
Not quite yet.
Okay, sorry. There I am. Alrighty. So our project area that we've been working on since 2015 is 3 coastal counties and 8 watersheds, from Lake Crescent down to Westport now. And the green are reed canarygrass, and the points and symbols on these 2 maps are 2018 to 2025. All species that we've been working on, and then just reed canarygrass on the right side. And the little blue circle is around Irely Creek and Irely Lake in the Upper Quinault. So our objectives are to protect climate, protect biodiversity, and grow really big trees for passive restoration. And we really don't accept the typical response to a lot of invasives, that there's just too many and they're out of control and there's nothing we can do. Because if there is nothing we can do, we can just kiss our cold water species, including salmon, goodbye. So we just say, get to work, get out there and hire lots of folks. And we've been developing programs to do that. And I'm not going to spend any more time talking about that here. But I invite you just to download the presentation or connect with me to ask questions. And I want to introduce Bob Vadas. Again, he's been an amazing partner in that he's a fisheries biologist, really focused on this endemic and landlocked population of cutthroat trout in Irely Creek and Irely Lake. And I've never had a fish biologist until I met Bob, who was willing to pay attention to invasive plants other than trees. So Bob has published a ton on this. The presentation is linked below there. And he has lots of written publications as well as presentations that are available. So I'm going to switch now to a slide that has escapement across years, and I think maybe it doesn't have the 2024 escapement in there, but Bob, would you like to make some comments about this?
Would it be possible to share my screen for a second to show the updated slide? Is that possible? No, probably not possible. Maybe in the panel, maybe at the end. But what I'll say about this slide is that the green stars are the population modeling that was based on data through 2012. There was 2 missing years that we modeled and showing at a time when the population was rising, and the model when we got back in 2015, the model was accurately predicting what we actually got. It stopped being accurate during the pandemic and in 2024, it was predicting that the population was going to be extirpated, and it didn't. And the reason it didn't, we believe, is because of the reed canarygrass control gave the population time to evolve to be more of a resident population, less dependent on the lake that was more frequently drying out since the turn of century. So probably climate change. So the 2024 data shows that an increase in the population, and maybe I can show that later. Yeah.
Great. Thanks so much, Bob. Yeah, we've gotten a lot of support from Olympic National Park. Bob's been working with the park since 1999 on this, and I've been working with the park since 2010 on reed canary Quinault. This was a poster that we did to develop a field study. And sorry, it's kind of blurry, but it is up on our website as well. You can pull it off and check it out there. We presented that in 2019 to the Salmon Recovery Conference. Been trying to build collaboration, cooperation, project integration with restoration sponsors around the coast on the Olympic Peninsula, both sides, on reed canarygrass for now since 2013. When I first went to Lee, and in 2010, actually, when I first was working in the Quinault. So it's been really hard to get any traction, to get a statewide program, to get research on reed canarygrass in cold water streams, to show the impacts on water quality, water quantity, fish populations. And Bob's study is super important for that. So we presented to the Society of Freshwater Science in 2021, and those were the partners we had with the project then, and they are still project partners now. So really grateful to everybody there. And there's Bob's slide again from the cover. And I'll just quickly go through these slides so that you can see some of the direct impacts on cold water ecosystems. It really fills them in. It really slows things down. It creates these deep mats of rhizomes that build way up out of the stream and trap finds. It basically makes wet areas into dry areas. And frogs and toads can live in reed canarygrass, but they cannot breed successfully in it. So that's a cutthroat trout that I pulled out of a ditch where it was stuck in super hot water and let it go in the Solduck River. He was really happy to get going. And so stream velocity decreases, increased stream temperatures, and decreased dissolved oxygen, and just a cascading effect on aquatic ecosystems, including on food webs. So we tried to do a BACI design, before and after control impact design in Irely Creek, and ran into some significant issues in both of our creeks. And this is where both of our study sites are. And one of the things that we have been trying to get going is this active response. So there's no, as I said, no research in Washington State or the Pacific Northwest on this species. And these are the questions we need to answer. And then we need to decide whether or not it is worth our time and money to save cold water ecosystems from this very aggressive ecosystem engineer. Our protocol is also available on our website, and we train in that. So to get an effective response, we've been trying different strategies throughout the last 13 years, doing the work, training people, doing protocols. We tried the study. We're expanding the study. We're going to be able to use remote sensing technologies, and we're going to be doing an intrinsic potential model project for coho and reed canarygrass. So we'll be doing a regional symposium hopefully this year, later on in the fall, and continuing to raise funds and hoping to publish. But we can't keep up right now, so just trying to figure out how to get the word out and get the money in and the jobs going. And that's an image of Irely Creek at the old log bridge from the east side to the west side. The bridge is down now, I think. I don't think it's been replaced yet. And that's Bob in the middle at the bottom there, and that's our a couple of our folks, Annika Fain and Marina Hine, studying. And you can see all the grass. I don't know if you can see my cursor, but that blonde green on top, blonde on the bottom, and those are the rhizome mats that are building up out of the stream and just filling in the stream. So we're really glad to hear from Bob that the that the cutthroat are seeming to pick back up again, because once they're gone, they're gone. And I just want to make some quick acknowledgments to everybody from 10,000 Years Institute who's contributed to this. Gino Lucchetti at the bottom and Stephen Stanley are part of the IPM project that we'll be doing ongoing. And then, of course, Bob and all of his volunteers. Joanne, I know, is on this webinar. And everybody from Olympic National Park, thanks so much for helping us and putting up with us. Catherine, for doing the lake control work that you've been doing. And then our funders have been Coast Salmon Foundation, the State WICRI program, the Surfboard program, Quinault Indian Nation, Washington State Department of Agriculture, and NIFLIF over the years. And we're doing satellite imagery for Scotch broom, which we will switch over to reed canarygrass pretty soon. And that's all I've got. Thank you so much. Sorry I went over there, Kim.
Oh, no problem. It's really interesting to hear what you guys are finding out there. And reed canarygrass is one that we battle here at Mount Rainier too. So it's always good to get more information and hear what other people are finding with it. So
it's totally doable. So I'd be happy to come over and do some training over there too. Thanks.
Yeah, would love to chat more. Okay. Yeah, thanks, Jill.
I'll stop share.
Okay, awesome. So our next presentation is going to be from Ryan Walsh. Ryan is a current graduate student at the University of South Carolina, who will be sharing some research that was conducted while an employee of the Makah Tribe. And it's going to be about the mortality risk for coho salmon landed in recreational troll fisheries using 2 different hook sizes. Thanks, Ryan.
Thank you, Kim. All right, I'm sharing my screen here. All righty. So thank you. I'm excited to share a study that investigates 2 different hook sizes in the mortality risk posed to coho salmon in a recreational troll fishery. The study was the efforts were led by Jonathan Scordino, an employee of the Makah Tribe, as well as our fellow colleagues at the Makah Tribe, Will Jasper, Dion Roche, and William Tyler. So to start off, salmon managers are tasked with balancing multiple priorities for salmon management. They have to consider economic, cultural, and recreational priorities to provide fishing opportunities for these 3 different realms, as well as ensuring that we're restoring healthy salmon runs and recovering depleted salmon populations. In Washington State, recreational salmon fishing is regulated with mark selective and size selective restrictions, and salmon under specific sizes and of wild origin rather than hatchery origin, which are marked with the removal of their adipose fin, they're required to be released back to the water. But this doesn't happen without the stressors and injuries associated with being hooked and landed. About a quarter of salmon hooked and released suffer mortality. So our research objectives, we aim to find a method that improves mortality from hook and release in recreational troll fisheries. And to do this, we looked at the 2 different size hooks and their effect on expected mortality risk for coho salmon in particular. And then we evaluated the drop-off rates and size selectivity of the 2 hook sizes evaluated to see if there were any additional benefits to regulating hook size in the fishery. This map depicts our fishing effort with each individual dot representing the start of a fishing set. We fished following Washington State's recreational troll fishing regulations in the summer of 2023 and 2024. We fished out of the Port of Neah Bay in Washington Marine Areas 4, 4B, 5. Here, this is depicting our 2 hook sizes that we employed, as well as our gear configuration. We used small and large. Our small hooks were of size 1/0, and our large hooks were of size 6/0. Our 6/0 hooks had a wider gape width or opening width in the hook as compared to the 1/0 hook, and our 1/0 hooks had a smaller gauge metal that they were manufactured with as compared to our 6/0 hooks, and we'll circle back to this later on in the presentation. Our gear configuration, we fished exclusively with hoochies as our lures, and ensured that we standardized the configuration of the hooks within the hoochies, where the bend of the hooks, whether it was a 1/0 hook or a 6/0 hook rigged on the lure, aligned with the tail end of the skirt of each hoochie. The hooks were then tied on to a monofilament leader and attached to a flasher, which was then attached to a fishing rod. Our study relies on the mortality risk produced by Wertheimer et al., where 6/0 J hooks were used in a commercial troll fishery that targeted Chinook salmon. So the table on the right-hand side summarizes the mortality rate produced by Wertheimer et al. For large Chinook salmon and for small Chinook salmon, with I color-coded our classifications of the risk of mortality, where we defined low risk of mortality as any body region where there was less than 15% mortality risk in the Wertheimer paper. And then in yellow, we have our intermediary risk of mortality, anything from 15 to 50% mortality rates. And then our body regions in red are our gill areas, which are our high expected risk of mortality, which is anything greater than 50% mortality risk. And the 9 different anatomical body regions described by Wertheimer et al. Are depicted in the image on the left-hand side. And this is our field guide for understanding where each body region is. So this table here summarizes the results of our study. We specifically looked at coho salmon and the effect of the mortality rates of the 1/0 and the 6/0 hooks. So in brief, we had slightly more fish, more coho salmon caught on the 6/0 hooks as compared to the 1/0 hooks. And between both hook sizes, the majority of fish were of legal size. While 6/0 hooks had a greater percentage or frequency of fish that were bleeding severely after being hooked and landed, these differences between 6/0 and 1/0 hooks for the frequency of bleeding was not significantly different. The main findings of this study are that for 6/0 hooks, we had a 2.8 times likelihood as the 1/0 hooks of hooking a coho salmon in a body area defined as either high or intermediary risk of mortality. And a little bit conflictingly, we saw that for our 1/0 hooks, they had a greater frequency of drop-off rates as compared to the 6/0 hooks, with 1.8 times as likely for a fish to drop off the hook before it was landed. Looking at the average length between coho salmon landed on the 1/0 and 6/0 hooks, there was not a significant difference, so there didn't appear to be any size selectivity between the 2 fishing hooks. So the management implications and future directions of this study. So I mentioned that we had conflicting results from the hook size effects on mortality rates for coho salmon. While the 6/0 hooks were more likely to cause direct hooking related mortality, our 1/0 hooks were more likely to cause drop-off rates and potential mortality of an unknown of an unknown frequency. I'll also briefly mention and introduce that we caught Chinook and pink salmon in addition to the coho salmon that we focused on here in this presentation. There didn't appear to be a significant difference in the hook size effect on mortality between those two species. However, our conclusions on the hook size effect for these two species was limited by low sample sizes. So while we don't make any suggested revisions to current hook size regulations in the fishery, we believe that our results can spearhead additional research to better inform an easily implemented regulation that can potentially improve salmon management strategies. These include a deeper investigation of mortality risk for other salmon species, such as Chinook and pink salmon, which we caught in our region, as well as verifying our assumptions. So we rely on the assumption that mortality rates from a commercial fishery that targets Chinook salmon using barbed 6/0 hooks, that those mortality rates would be equivalent to those observed for a recreational fishery that's catching coho salmon on both barbless 1/0 and 6/0 hooks. And then lastly, we noticed the increase in drop-off rates in the 1/0 hooks as compared to the 6/0 hooks. And this can be due to the difference in structural integrity associated with a smaller gauge metal used in the manufacturing of a 1/0 hook as compared to the 6/0 hook. So investigating the difference in between the two size hooks using the same size or the same gauge metal would help to determine if drop-off rates are associated with hook size directly or if structural integrity of the hooks is playing a role there. I just want to thank my collaborators on this study and mention that the project was made possible by funding from the city, which is a program of the Karen C. Dreyer Wildlife Health Center in the School of Veterinary Medicine at the University of California, Davis. And then we had many volunteer anglers throughout our study, which made this effort possible. Thank you all.
Thanks, Ryan. Really interesting to hear what the results of those studies were. Thanks so much for sharing with us. Okay, we have our final lightning round presentation coming up. And I believe it's two folks joining us for this next one, Dr. Ian Miller and Dr. Steve Fradkin, who we met earlier during one of our other lightning sessions. And they're going to be talking about deploying chronologues in Olympic National Park to support coastal research and interpretation. Thanks a bunch.
All right, thank you. Can you confirm that you can see my slide?
I can, and I can hear you. Looks like you're all good to go.
Excellent, thank you. I actually don't know if Steve's going to join, but hopefully I will suffice. My name is Ian Miller, and I'm the Coastal Hazards Specialist for Washington Sea Grant. I'm based in Port Angeles, and my co-author, Stephen Fradkin, and I are going to attempt to summarize some of what we think we've learned using chronologues, which are a system that asks users to submit photos of areas of interest, which in our case is the shoreline around Claylock in Olympic National Park. So going to start with just a bit of orientation about Claylock. This is on the coastal strip of Olympic National Park, and it's really an important coastal area for a whole variety of reasons. It sits right along Highway 101, which makes it easy to access. It's also the home of a large National Park Service campground, as well as Claylock Lodge and some 30 associated cabins. Those are part of a concession. And so it's a heavily used area and also a key area for providing access to, this gorgeous stretch of shoreline along the coastal strip of Olympic National Park. Claylock, though, presents some management challenges associated with erosion of the coastal bluffs that back the beach. And the photograph here is of those coastal bluffs looking up from the beach. And those threaten park infrastructure, like the access trails to the beach and the cabins at Claylock Lodge. Last year, in fact, National Park Service removed 5 structures from clay lock because of this erosion. And this erosion has also prompted the development of research and monitoring programs of various kinds throughout the years, including one that I run and presented on at last year's Science Days. And those are intended to better understand coastal processes in general and erosion in particular. And in general, those monitoring programs that we do rely on data sources collected relatively infrequently, maybe every couple of years in the case of aerial photos or LiDAR, or every few weeks at best, or at best in the case of field survey data that Steve and I collect, for example. So in the hopes of getting more frequent insights about change at this site, in early 2023, Steve and I launched an effort to place 5 chronologue stations at Claylock, which we ended up installing in October of 2023. And so they've now been operating for about a year and a half. Those stations were, as far as we know, the first chronologue stations in Olympic National Park. And these were also placed with some support from the NSF-funded Cascadia Copes Hub. So chronologues are a photo submission system, so sort of like MyCoast or CoSnap, if anyone's familiar with those programs, kind of similar in the sense that they ask community scientists, users to submit photos. And to date, we have about 700 photo submissions from the 5 stations at Claylock by about 500 users. So in the case of chronologues, users are asked to take a photo from a fixed cradle sitting on a post. So I've got this arrow pointing out the cradle. So we get to select the field of view, and it's consistent amongst users. And then they submit it via email to add to our image database for that location. So I'm going to spend the rest of my time going through 4 sort of takeaways from our experience so far. And the first is that the system has worked in the sense that it provides us with new insights about coastal processes. So I want you to pay attention to the logs on the beach in this photo. And this was submitted in December in 2023. And this, by the way, is a field of view from one of those chronologue stations. And I'm going to switch to a photo collected 4 days later. So compare it to this one. And in this case, these observations have provided us with a new understanding for basically how mobile that large wood is that's commonly seen along the shoreline of Olympic National Park and certainly along Claylock, which may have implications for habitat, erosion rates, and even access on this section of the coast. So the second thing that we think is a key takeaway, something that is a real benefit, is that like a lot of the other systems out there, Chronologue provides a really nice exchange with the user. We get the observations for our area of interest, and the user, though, gets an immediate response back via email with interpretive information that we provide, as well as their photo integrated into a time lapse from the site. And so this provides some interpretive potential that would otherwise be very difficult to deliver to these kind of really engaged users at Claylock. So for the third takeaway, I just want to emphasize again that this is all email-based. Most other systems like this, I mentioned MyCoast, I mentioned CoSnap, are app-based systems. But we think that chronologues, which are configured around email, are really well tuned for applications in remote areas like relatively remote, where cell signals aren't fantastic always, and for users that may be coming from all over the world and may not be interested in or able to install an app, especially while they're standing there kind of waiting to use or engage with the system. And then finally, deploying these 5 stations around Claylock has really helped us to understand use patterns, which we have found very widely. So location, as is the case with real estate markets, really matters. We have 4 stations, I've sort of highlighted those on the screen here, that are generally attracting roughly the same amount of use. We have 30 to 50 users on average at each of these sites over the last year and a half, submitting on average a photo every 5 days or so. And then by contrast, we have one of our stations that sits right on a trail leading down from the parking lot of the campground. And it's really kind of right in people's way as they move down that trail. that really swamps the others. And in this case, we've got 406 photos contributed from this station, so almost 10 times as many photos on average by 385 contributors. So a lot of use at this station. So in this case, we think that front and center placement of that chronologue station is really key, and are now experimenting with different strategies for trying to drive users using this station to some of our other stations so we can get heavier use on those other stations. So in sum, our experience to date with deploying chronologues at Claylock, we feel has been pretty positive. I should have noted as well, these are relatively cheap systems to run. Each station costs about, on average, $100 a year to be maintained by Chronologue. And we think we're also really starting to gain some insights that will help us to fine tune their use in the future. So with that, thank you so much.
Awesome. Thank you, Ian. Thanks for sharing all that information. Really neat to hear about. Okay, well, that ends our last round of lightning talks. So we have a couple minutes left for question and answers. So we'll do that. And then we have our closing remarks from Greg at 3:50. So I think we have a couple of questions we can start out with that we have down in our chat, which it looks like, Lindsey, you might have already jumped on and answered some of those, which is awesome. Let's see. So talking about drones with mapping, and then also the feasibility of screening out rocky areas that led to overpredicting social trails. Looks like you already answered those, but did you want to share anything else about that, Lindsey, for the question and answer session?
I can just briefly go over what I said, so people don't have to read it. Greg asked if drones could help with more precise mapping. And yeah, I mean, drones totally could help with collecting imagery, but when working in national parks, it's pretty hard to get a drone permit. It's like essentially impossible even for research grants, unfortunately. So hopefully we'll get some better imagery soon. And John asked about screening out rocky and buried areas that led to overprediction. And a lot of the areas that we're looking at that are really highly impacted do have a lot of rocky or barren areas that are really close by, which is a challenge for a computer to distinguish between those two. I also mentioned I did rerun the model for a smaller subalpine area in Paradise, but it was just in the office, so I wasn't able to field validate it because the snow had already started falling. So hopefully in the future, me or someone else will be able to get out and continue looking at that and continue to model social trails at Paradise.
Thanks, Lindsey. And then a question here from Beth Fallon. Deneb's, how much were the differences in species at the restoration site versus the surrounding sites driven by planted, salvage species differences?
Yeah, so that's sort of one of the questions that I had and was looking at. I did have access to which plants were planted, and sometimes it seemed to match, but also sometimes it didn't. Like that sandwort that was dramatically different, that there's lots of sandwort in the surrounding meadow and not very much in the septic meadow. It was one of the most heavily planted in the septic meadow, so that might be more of a survivability. The factor of the yarrow is pretty heavily planted, so that one might be due to planting. So I think there might be a couple of different dynamics going on.
And I had a follow-up question for you as well. I was curious with those restoration sites, if you guys did any additional like seeding or mulching with any of those sites as well, or is it mostly like the plugs and the salvage planting?
Yeah, it was mostly the plugs and the salvage. There was some seeding. Specifically, there was one section that was sand fill with jute netting that had plugs planted in it, but I know some seeding was done in there. And I do have the numbers for the seeding, but it was much specific spots.
Okay, awesome. And it looks like, okay, we have a couple more questions coming in. For Ian Miller, do you have other community science initiatives that Chronolog fits into? Or is Chronolog your team's first foray into creating a community science program? What are your priorities around that?
Well, Sea Grant, who I work for, has a number of community science initiatives. So I would encourage, anyone to go check out our website. Probably the one that comes to mind, at least relevant to a previous Science Days talk in the last session, was Emily Grayson, who runs our green crab program, and they have a considerable community science component associated with that program. And we also run what's called the King Tides program, which is a program that asks users to collect photos of flooding events. That's one that we're actually a partner on the My Coast app, which users use to submit those photos. This is my first experience with using chronologs in particular, although there are other applications on the Olympic Peninsula that have emerged since that time. The Lower Elwha Clown Tribe placed one, for example, on the former, pointing at the former lake bed of Lake Aldwell, so associated with the Elwha Dam removal, that is just off the highway where you cross the bridge if you're heading from Port Angeles west. And then I believe there's the national park may have placed one up at the former upper dam site, the Glines Canyon Dam site, directed at the lake bed, the upper lake bed, Lake Mills. But I'm not sure if that actually got placed yet. And we've talked about other applications. There's a similar photo-based system. It's a co-snap system down at Washaway Beach at North Cove, north of Willapa. So these kind of systems are emerging for coastal applications and other applications quite rapidly.